So in this topic, we're going to look at gene sequencing. DNA sequencing is the process of working out the order of bases in a strand of DNA. In 1980, Frederick Sanger won the Nobel Prize for working out a method of how to do this. The first organism to have its genome sequenced was Phi X174, a bacteriophage, a virus that infects bacteria. Since then, sequencing techniques have improved enormously. Before we look at the process, you should know about a special type of nucleotide that we need to use. These are what we call Terminator bases. They lack the OH group on the carbon 3 of the deoxyribose. They therefore cannot bind to the next phosphate group on the following nucleotide, as no phosphodiester bond can form. Therefore, this terminates the strand extension during PCR if one of these terminator bases binds. There are terminator versions of all the 4 bases in DNA. So, how do we do DNA sequencing? Well, step 1 involves getting 4 labelled test tubes, A, T, C and G. And into these test tubes you put fragments of the DNA sample that you want to sequence, that you've cloned using PCR. You put plenty of free DNA nucleotides to be able to make more DNA, and you put in DNA polymerase. Then, in step 2, you're going to add 2 further very special ingredients. You're going to add the relevant terminator bases to each tube. So I've labelled these as A-, T-, C-, G-. These are the special bases that will then stop the DNA replicating at that point because it can't form any further phosphodiester bonds. So we put the relevant terminator base into tube A, A-, and the relevant one into tube T, T-, etc. We also put a fluorescent primer into each tube, a different fluorescent primer to each tube that allows us to label these DNA fragments and therefore detect them later on. So we put a blue one into A, yellow to T, C is green and G is red. In step 3 we run PCR and we get the strands to replicate. Okay, and the DNA polymerase binds to the primer and makes the complementary strands using the free nucleotides as you'd expect. But at about 1% of the time, a terminator base will bind and the sequence stops. It won't always stop at the first chance, as there are also normal nucleotides, but over time you will get all the possible points at which it can stop occurring due to those terminator bases binding in each of the 4 tubes. And this will happen in the equivalent way in all 4 tubes. All these fragments then get mixed together into one tube, and they are then separated by size in a process called capillary electrophoresis. A laser shines through the capillary tube and detects those fluorescent markers as it crosses, and the smallest fragment moves first, and then they go in order. So it allows to then generate an order of what those bases were based on the fragments. And this gives us the full sequence. Using the genetic code, we can then work out what the amino acid sequence should be. We used to think that each gene coded for only one protein, but we now know that we have these 20,000 genes, but due to gene splicing, it's thought that these can make up to 500,000 different proteins. As we start to learn more and more about the genome, we begin to understand just how complex it is. The hope was that with the knowledge of this human genome, we'd be able to identify specific mutations that cause human diseases and then treat them better. We have identified some single gene diseases like haemophilia, cystic fibrosis, but we haven't managed to successfully cure them yet with gene therapy. The reality is a little more complex, as most diseases have an environmental component as well, and many of them are actually a result of a number of gene variants, not just one particular mutation. However, big projects are now underway to sequence hundreds of thousands of patients with complex diseases such as cancer and diabetes, and then computers can process this huge amount of data to look for genetic patterns.